Hello, everyone, and welcome back to the Simcyp technical webinar. My name is Xian Pan. I'm part of the translational DMPK group at Simcyp, and I will host the session today. So today's topic focuses on the long-acting injectables module in Simcyp Simulator. Before we start the talk, I'd like to kindly request that everyone stays muted during the presentation. Should you have any questions, feel free to raise your hand or post your query in the chat box. So we have a dedicated Q&A session at the end where we will address them all. So we're recording the presentation, which will be available on YouTube shortly. Next slide, please. It's my pleasure to introduce today's speaker, my colleague Naresh Mitapalli. So Naresh is a senior research scientist in Mechanistic model group of Simcyp. He has the academic background in pharmaceutics and pharmacy. Naresh has been with Simcyp for about 5 years and leads the long-acting injectable project. I'm glad to have him with us today. The talk today will cover an introduction to Simcyp's polymer-based injectables and implant models. We will also show you how to parameterize the model and followed by a case example. Without further ado, I will hand it over to Naresh. Over to you.
Thank you, Xian, for the introduction. I just want to keep my video off to save some bandwidth. I don't know, it's not working. Okay, let's go. So let's start the presentation with a brief introduction about long-acting injectables. First of all, thank you very much for attending this session. So I just wanted to start with introduction to long-acting injectables and what type of formulations are there in the market. So typically long-acting injectable drug products release drug from few days to months. The most common routes are subcutaneous, typically arm or abdomen. For intramuscular administration, it can be deltoid, gluteal, or vastus muscle. Based on the formulation point of view, these products can be classified into different categories. There are solid implants, which can be biodegradable solid implants or non-biodegradable solid implants. Some products are suspensions based on drug and prodrug technology. Some products are biodegradable microparticles, which are based on PLGA. There are also oil solutions of drugs and products available in the market. In addition to the most common route, sometimes other routes of administration also reported in literature and there for the administration in patients. For example, Ozurdex, which is nothing but dexamethasone solid implant, it is for intravitreal administration. So before going into the actual model, I just wanted to talk about Simcyp timeline of development for different models. In version 22, we released solid implants and in situ gel forming implants model. This is available in IVRT module as well as in vivo simulator. Second model, it was released in version 23, which is aqueous suspension based long-acting injectable model. This is for handling the suspension based long-acting injectable drug products. In version 24, we have made some updates to the long-acting injectable models. So currently we can handle solid implants, in situ gel forming implants, and suspension-based long-acting injectable drug products. So before going into the actual case study, I would like to discuss and talk about the model that we have in the Simcyp simulator, about the model structure and background. So this is the model structure that we have in the Simcyp simulator, where we have a solid implant in contact with the release media. So when you look at this model, this is a model developed to explain the drug release from PLGA-based solid implants. And this is a mechanistic model. And when you look at the mechanistic model, it considers different processes that are known to happen in case of solid implants when they are in contact with the release media. There are empirical release models available out there. However, when you compare empirical release models with mechanistic models require different source of parameters compared to the empirical release models. But they can explain different scenarios. For example, if you want to change a particular critical quality attribute and want to know what is the impact of that critical quality attribute on drug release and in vivo clinical PK. So you can do that with a mechanistic model confidently, whereas with empirical release models, they can be used to explain the data to that particular case, particular design space. So for this model development, for this polymer-based biodegradable injectables and implant model development, we have considered different processes which are known to happen in case of PLGA-based implants. So as I said, the solid implant in contact with release media, when the implant is in contact with release media, there is an influx of water slash release media into the solid implant. Because of this influx, the solid implant can be divided into two compartments, wetted compartment and unwetted compartment. So we mechanistically handle this process in the model. There is a non-catalytic hydrolysis of PLGA, which is nothing but in the presence of water, PLGA undergoes degradation. This we call it as non-catalytic hydrolysis of PLGA. When there is a degradation of PLGA, the PLGA chain lengths would decrease over time, and the produced separate chains, they would release H plus ion, they would release H plus ion from carboxyl terminals. So these carboxyl terminals released H plus ion would participate in further degradation of PLGA, which we call it as non uh, autocatalytic hydrolysis of PLGA. So this autocatalytic hydrolysis of PLGA involves participation of H plus ion that is produced from the initial degradation followed by uh, autocatalytic degradation of PLGA. So which means the PLGA degradation accelerates because of its own products. So that's why it is called autocatalytic degradation of PLGA. So because of this autocatalytic degradation, non-catalytic degradation, the solid implant microstructure would change from solid structure to more porous structure, and there would be dissolution of oligomers. Oligomers are nothing but small chain PLGA structures, so they would diffuse out of the delivery system when they reach certain chain length. For example, if the chain length is less than 16, we can assume that the polymer oligomers can diffuse out of the solid implant structure, and there would be mass loss from the solid implant into the release media. So we consider dissolution of oligomers. We also consider dissolution of drug in the pores. So when the pores are wetted with release media, there is a dissolution process happening. So we handle that dissolution using modified Van Flanagan equation in the model. Also, there is a liberation of drug because if you look at the solid implant structure, there could be pores and there could be matrix. So in order to release, in order to dissolve, it first have to untrap from, it first have to liberate from the matrix structure to the, porous structure. So that we call as liberation of drug from matrix to porous structure. So this we consider in the model. So in case of in vivo model, we have interstitial fluid compartment that is in contact with the solid implant. And this interstitial fluid compartment connects with adipocyte compartment in case of subcutaneous tissue, or muscle tissue compartment in the case of intramuscular injection. There is a transfer of drug happens based on the physicochemical properties of drug. For example, partition coefficients. If the partition coefficient is high for adipocyte compartment, there is a possibility of local depot formation in the adipocyte compartment and slow release from this adipocyte compartment back into the interstitial fluid. Also, there is a connection between interstitial fluid compartment to the systemic circulation. So this we handle using diffusion. We need additional parameters like partition coefficients and diffusion coefficient values in order to parameterize the in vivo model. So this is just to show you what happens, what are the sequence of interacting events when a solid implant is in contact with the release media. So initially implant is in contact with the release media. Because of this, implant compartment is divided into unweighted and weighted compartment. And because of the hydration, there is a production of acid polymer chains because of non-catalytic PLGA degradation. This acidic polymer chains would participate in further degradation of PLGA, which we call it as autocatalytic degradation. Because of autocatalytic degradation within the implant, there is a pH drop in situ. This will go to acidic side because of accumulation of H plus N due to the degradation of PLGA. So next step is diffusion of oligomers and drug. When the chain length of oligomers reaches a critical value, they would diffuse from the system inside to outside. This would lead to a change in mass loss and macro level. At macro level, there would be a change in polymer. There would be a change in porosity value. We also consider the dissolution of drug, as I previously mentioned. Drug dissolution happens at pore level. So we consider the pore surface for this dissolution process. And these are different processes happening simultaneously and handled mechanistically in the model. And one process does not limit another process when it comes with mechanistic handling. So in order to parameterize this mechanistic model, we would require different parameters that are coming from different sources. We can broadly classify them into 4 different categories. They are drug-specific parameters, polymer-specific parameters, formulation-specific parameters, and system-specific parameters. Also in the model, we assume linear lactate-lactate bonds and glycolide bonds. So this is our basic assumption in the model. So we are not handling lactate-glycolide bonds as a polymer chain. For the purpose of computations and simplicity, we are currently considering there would be a lactate chain and there would be a glycolide chain, and polymer is distributed in these 2 forms. So example for drug-specific parameters are molecular weight, solubility, pK information. So this information would go into the model. For in vivo case, we need additional parameters like partition and diffusion coefficients. Polymer-specific parameters are hydrolysis rate constants. We need lactate-lactate hydrolysis rate constant, bond degradation constant. Also, we need glycolide bond hydrolysis rate constant. We also use diffusion coefficient of oligomer in the model because oligomers diffuse out of the system. So because of this only, there would be a mass loss within the solid implant. Formulation specific parameters are like porosity of the system, average pore radius, initial releasable fraction, implant dimensions. So these can come from the characterization of solid implants during formulations development in the R&D. System parameters, we consider release media volume, experimental settings like pH of the media, viscosity of the media. So they would be utilized for running the simulations and model development. In case of in vivo, we consider local tissue composition and blood flow as system parameters and use those values in the model for running the simulations. So before going into the case example, I would like to quickly show you how the user interface would look like and where the parameters are incorporated into the user interface. So this is Simcyp version 24. Here you can see we have a IVRT module. As I previously mentioned, with IVRT module, we can run in vitro simulations for solid implants. So here you can see within substrate, we have physchem information where user can input molecular weight, intrinsic solubility, if the molecule type is monoprotic, diprotic, they can do that. We can also incorporate PK information, drug particle density. From the information like molecular weight, we can calculate the molecular radius. So this is the screen for inputting different parameters, different category of parameters like drug diffusion coefficients. This can come from a calculation. Uh, so if we just input the molecular weight, we can calculate the diffusion coefficient from predictors. Polymer parameters, most of the polymer parameters are supplied by default, let's say molecular weight of oligomer 1, oligomer 2. So these values are already inputted into the screen. So these are all different default values. As I mentioned, we use diffusion coefficient of polymer. What is the critical chain length for oligomer dissolution? Only dissolved oligomer can only diffuse out of system. Most important thing is consideration of critical quality attributes for modeling. So we consider critical quality attributes of formulation like lactide to glycolide ratio, contact angle of implant, fractional volumetric porosity, initial releasable fraction, what is the pore radius of system. In addition to that, we also consider implant parameters like length, radius, mass. On the top of that, user can input molecular weight distribution. So they can define the distribution as a normal distribution, or if they have values, they can input discrete distribution as well. So in terms of in vitro design, we can specify the dosage of formulation, volume of solvent, viscosity of solvent, surface tension, etc. So this is just to give you a brief idea how the user interface would look like before going into the actual case example. So the case example that I'm today going to discuss about is Ozurdex. So this case example related data / information is taken from a publication from University of Texas at Austin. So they have manufactured dexamethasone reverse engineered implants. They also characterize the commercially available Ozurdex implant, which is intended for intravitreal administration. So this implant is expected to release drug for up to 6 months. They have used hot melt extrusion as a process for manufacturing the implants. If you look at the composition of this implant, the implant has dexamethasone. It has 50-50 lactide to glycolide ratio polymer. Acid terminated polymer is about 30 percentage, whereas ester terminated is 10 percentage. So in the model, we do not explicitly consider ester terminated polymer. So we can consider the effect of ester terminated polymer by accounting for contact angle. So if ester terminated polymer is there, you can expect the contact angle value would increase compared to acid terminated polymer. We can do that if we have a measurement, but for this particular case example, we did not consider that. So apart from this publication, there is another publication, Bhagwat publication. In this publication, they have reported the release of Ozurdex commercial, which is a commercial product. They have reported the release of this commercial product. So for the model development, what we have used is we have used Ozurdex release data, and we have incorporated all the information that is available from this publication. For example, porosity value, molecular weight of PLGA, lactide to glycolide information. So most of the information is available from uh, University of Texas publication. So in order to recover the observed data, so we have optimized the model parameters related to autocatalytic degradation of PLGA. So we have two different parameters, ionization proportionality constant and deionization rate constant. They are going to influence the autocatalytic degradation process. So for the model development, we have optimized these two parameters. And once the optimization is done, we validated this approach with a different profile that we have from Bhagwat et al. So where we have seen by changing the profile, by changing the experimental conditions reported in Bhagwat et al. For marketed implant, we recovered the data well. So I'll show you the results. So just to show you what is the impact of different parameters, like we have immediate releasable fraction slash initial releasable fraction, ionization proportionality constant. As I mentioned, ionization proportionality constant, it is going to impact the autocatalytic degradation of PLGA. So because of that, when the value is increased, there is a more degradation. Because of that, you see more drug release. Initial releasable fraction in the model is it's a bit a tricky parameter. So it actually conditions the drug distribution within pore space versus matrix space. So this is not exactly what we assume when we listen to the name initial releasable fraction. So that's why, like, when the value is changing from 0.003 to 0.3, we do not see like a massive increase in drug release in case of sensitivity analysis for immediate releasable slash initial releasable fraction. Again, deionization rate constant, it is going to decrease the autocatalytic rate process. So when the value is high, you can see a delay in drug release profile. So lactate to glycolate ratio, this is a critical quality attribute. Compared with glycolate bonds, lactate bonds are tend to degrade slowly. So because of that, when there is a lactate content increase in the formulation, you see a shift in release profile from left to right, which means there is a delay. We consider pore radius in the model. As I said, this is the impact of pore radius for this particular simulation. Pore radius can impact the diffusion of H plus ion, diffusion of oligomers and drug. So this is the impact of porosity value. Again, the same logic applies here. The same logic as that of pore radius. So if you have a non-porous system that retains H plus ion, it would degrade the system, causing more heterogeneous degradation of PLGA and drug release. If the porosity is high, then you can expect some delay in drug release for given set of parameterization. So this is the profile that I'm talking about with Bhagavat et al. Study. So here you can see we just replicated the replicated the simulation with experimental conditions reported for Ozurdex implant and how the model is performing. So visually you can see the model is able to capture a profile that is reported in a separate study. So in addition to that, we also have reverse engineered implants data. So we have incorporated that information. You see there is a difference in PLGA molecular weight here. In case of reverse engineered implant, we just incorporated that information without changing any parameters. And this is how the model is capturing. In addition to the drug release profile, we also have information on polymer mean molecular weight profile over time and pH in solvent compartment. With respect to these two profiles, the model is currently performing okay. We are planning to address this discrepancy between observed data and simulated profiles in future. So with that, I would like to conclude this presentation on PLGA-based solid implants model. So where I have discussed the solid implant model, its parameterization, and Ozurdex in vitro release model development. The future work will focus on increasing the prediction accuracy for molecular weight and pH profiles. Thank you. I would like to take any questions if you have.
Thank you, Naresh. Now let's move to the Q&A session. We're happy to hear your thoughts. You can either raise your hand or type the question in the chat box. We still have a few minutes to go through a couple of questions. While we're waiting for them to come through, I'd like to ask a question. So in the shared case example, you have optimized the two parameters to recover the observed data. So my question is, how do you determine which parameter to optimize? In another word, which parameters are most sensitive in this context?
So in this particular case example, what we have noticed is ionization proportionality constant is sensitive. I mean, it's going to cause most effect on the release profile, shape, as well as magnitude. So we use sensitivity analysis for determining the ionization proportionality constant range. So in general, what we suggest is once you have the experimental data with one set of experimental data, perform a sensitivity analysis to understand the parameters impact, then go for optimization of those parameters for that set of data, then validate it by changing the critical quality attributes for a different release profile.
Okay, thank you. I think we have one question, and I think Tycho, you have your hands up first. You could unmute yourself if you want to ask questions.
Yes, thank you. Can you hear me?
Yes.
Yes, I can hear you.
Yes, thank you, Naresh, for a very nice presentation. I was curious, coming back to the ratio of lactide to glycolide. I think you had the release profiles shown. I was curious, were any of the data verified with measurements? I think you had one where you changed the glycolide level to 0%. And I was curious when there was any data for that. I mean, in theory, it makes sense, but if you wanted to use a model like this for regulatory filing, right, we would probably need to bracket this plot here on the left. I guess the yellow one, the yellow line is 0% glycolide, right?
It's 100%. Yeah, it's 100% lactide, which means 0% lactide. So just to give you the background, so it's just to show you the sensitivity analysis for this particular Ozurdex reverse engineered implant. So if we have data, then we can definitely validate the model with changing the lactide to glycolide ratios in theory. Yeah, there is a publication. In this publication, we have reported for IVRT case examples where we have different lactate to glycolide ratios and how the model is performing with the published case examples. Having said that, when you are manufacturing an implant, even though you change only material attributes like lactate to glycolide ratio, it is expected to cause some change in critical quality attributes, macro critical quality attributes like porosity. It can change the drug distribution within the implant, so which in turn can impact initial releasable fraction. So I think, in my opinion, what regulators are expecting is they wanted to understand, how the model is performing for your particular case example using prospective in vitro release studies. Let's say you manufacture a different implant with different lactate to glycolide ratio, and you study the implant thoroughly for characterization. You measure the porosity, you measure the molecular weight after manufacturing. So the more the data would go into the model, the more the confidence you can get. But at this stage, we have some examples in the IVRT paper that we published last year.
Yes, thank you.
Thank you, Tycho.
Thank you. We have one more question, which is from Anna. Thank you for the presentation. Could you repeat again how the ester versus acid PLGA ends would affect the model?
Yeah, in the model, we do not explicitly consider ester-terminated polymers. We can indirectly account for that by incorporating the contact angle. Like we have a specific parameter which is called contact angle. This contact angle is going to influence the wetting process. So if the contact angle is high, you can expect a slow wetting process. So that way we can currently account for that. Probably in future iterations, we would try to account this terminal ester group impact on degradation of PLGA. So it would be like for future improvements. Hope that clarifies.
Yeah, we get another question from Andrew asking regarding the initial release fraction. Why does it look like it only changes lag time to drug release? Is this because you're modeling a poorly water-soluble drug like dexamethasone? Would we expect for a higher solubility molecule a much more significant and a faster burst release?
Yeah, that can be partly true. Dexamethasone is it's a hydrophobic structure. Uh, it's a hydrophobic molecule, and the drug loading within this system is, if you look at the drug loading, it's around 60%. With that 60% loading, solubility is playing a role in this delay in drug release profile. Yeah, for hydrophilic drugs, when there is a change in initial releasable fraction, which means if you distribute more into the pores, if the system is wetted quickly, then you can expect a faster, release of drug because of this wetting process and subsequent drug release. That observation is correct. Yeah.
Okay, thank you. I think that's all our questions for now. If there are no further questions, we will now conclude today's webinar. Thank you, Naresh, for the talk about long-acting injectables and the Simcyp simulator. And thank you to everyone for joining us, for your attention and also active engagement. Should you have any additional questions later on, please don't hesitate to get in touch with us. And as you can see here, our next webinar is already scheduled on the 30th of October. So this is another webinar about a virtual bioequivalent module. And David will give you an example on that. It's a case based on the case example. So if you're interested in it, please register through members area. Again, thank you everyone for joining us today, and we look forward to seeing you soon. Wish you all a lovely day and goodbye.